Hello, I'm Yi Liu, a grad student from the Jackman Lab at Stanford. Today, I'd like to introduce our recent study investigating interpretable subspaces in brain-wide neural activity. I apologize for not being able to attend the workshop in person due to personal reasons, but I hope my talk could still evoke some thoughts and discussion, even though it is virtual. In recent years, progress on recording techniques has enabled large-scale simultaneous recordings of neural populations. For example, with the NeuroPixels probes, we are able to record hundreds to thousands of neurons at the same time. And by inserting multiple probes at the same time, we are able to perform simultaneous recordings of neural populations from multiple brain areas. This opens up possibilities of studying computations across multiple brain areas. But this also poses a computational challenge. How should we analyze populations of neurons in multiple brain areas? There are existing statistical methods that analyzes the communication between brain areas. For example, there is the canonical correlation analysis, or CCA, that finds pairs of axes so that the projections onto the axis have maximized correlation. And there's also the reduced rank correlation, which finds subspaces in the source area so that the variance explained by the neural activity within the subspace is maximized in the target area. These methods deal with the challenge of analyzing two high-dimensional data by finding low-dimensional communication subspaces in the neural population. Such dimensionality reduction makes it easier for us to examine the part of neural dynamics that is related to between-area communication. However, these subspaces usually suffer from the lack of interpretability, since they were fitted to mathematically maximize some statistical measures, and it is hard to understand what these subspaces encode. In comparison, within the neural activity in a single brain area, previous studies have shown that there are subspaces that encode meaningful variables, which makes them very interpretable. This is an illustration of neural dynamics in the high-dimensional activity space. The thinner lines indicate single trial neural dynamics, and the thicker lines is the trial average mean. The two planes represent two different subspaces, and therefore we can calculate the projection of the neural activity onto these subspaces. So it matters a lot which subspace we are looking into, since different subspaces could have different dynamics, different decodability, different temporal stability, etc. And therefore, in this study, we'd like to utilize what we have known about the subspaces in single areas to inform our thinking on analyzing multi-regional dynamics. We hope that by doing this, we will be able to understand more about what is encoded in the multi-regional dynamics. To look into this question, we start from looking into three aspects in this talk. First, we'll look into the importance of subspace-specific analysis. And second, we'll try to interpret the relations between subspaces. And specifically, we hope to interpret the subspaces that usually lack interpretability due to their statistical nature. And finally, we're gonna look into the capability in subspaces of predicting perturbation effects by utilizing the perturbation trials we have in the dataset. So the dataset we are using is the mesoscale activity project dataset. Here, the task structure is as follows. Each trial starts with a sample epoch where the mouse is given auditory tones that can either be high pitch or low pitch. The tone was repeated for 3 times, and the sample epoch is followed by a delay epoch. After that, there is a go cue, and after the go cue, the mouse is supposed to lick left or lick right according to which auditory tone it heard during the sample epoch. From the previous studies, we knew that the anterior lateral motor cortex, or ALM, is critical for planning directional licking. We also knew that there are these subcortical regions that are anatomically connected with ALM. And while the mouse is performing the task, we used up to 4 neural pixels probes to record the neural activity from different brain areas at the same time. And we also have some trials with perturbation, which I'll mention later. Meanwhile, we had video recordings of the mouse so that we have an idea of the movement of the mouse. This is the recording from one example session from multiple brain areas. For more information about this dataset, I'd be happy to talk later about our recent paper referenced in the bottom right corner. Now, let's start from the first question. Why is it important to investigate different subspaces? As a first step, we need to identify the subspaces of interest. Previous studies have found the coding subspaces that maximally encode choice and trial type, which is the instructed lick direction. For example, for the choice coding subspace, it is calculated as the difference between the main activity in lick right trials and lick left trials. And the trial type coding subspace is similarly calculated, but this time the trials were grouped according to their trial type instead of choice. And in addition, we also used canonical correlation analysis to find the between area correlation subspace, or BACS. CCA finds pairs of axes in the population activity space in each brain area so that it maximizes the correlation between the two areas. As I mentioned before, reduced rank regression is another method that finds communication subspaces between areas. But for the sake of time, we are going to focus on the results using CCA today. Furthermore, our video recordings showed that mice made uninstructed movements during the task. And therefore, we fitted another canonical correlation analysis between the neural activity and the video embedding, which I'll mention later. This finds the subspace in the neural activity that encodes movement of the mouse, and we call it the motor subspace, or MS. So once we have identified the subspaces of interest, the most straightforward thing is to explore the dynamics within the subspaces. Here, we project the neural activity of ALM onto the choice coding subspace. We're skipping the trial type coding subspace since time is limited. So this is the sample epoch, this is the delay epoch, and the solid line is the goal queue. Here, the red and blue traces are the main projections across trials within lick left and lick right trials. And the gap between the two traces indicate the decodability of choice. We see that as we approach the goal queue, the decodability of choice also increased along the CD choice subspace. So how could dynamics look like in the other two subspaces? It would totally make sense even if they look very similar to the coding subspace. But instead, we found that although the dynamics within BACS and MS are epoch dependent, they have little selectivity as indicated by the little separation between the two traces. We see that the dynamics is very different across different subspaces. Now let's focus on the choice coding subspace and take a closer look at the decodability. Here is an example session, and the trials have been grouped according to the instructed left or instructed right trials. Each column here is one trial, and we are showing its trial type and choice. And in the middle are the choice decoding results from each of the brain areas recorded in this session. We see that the choice prediction from the 3 areas are often very correlated, and specifically, these areas tend to make error predictions at the same time. To quantify this correlated decoding, we calculated the agreement ratio, which is defined as the empirical probability of areas having agreed decoding divided by the expected probability, assuming that these areas are independent. The baseline of the agreement ratio is 1 when the 2 probabilities are equal. And in this example session, the agreement ratio is about 5. Here we're showing the summary of all the area pairs consisting of ALM and another area. The orange bars are the choice agreement ratio calculated at the late delay epoch, and the blue bars are child type agreement ratio at the sample epoch. We see that all these values are well above 1, which shows that the coding of task-related variables is highly correlated between brain areas. So we just saw that the dynamics along coding subspace is very correlated. So will the BACS also find the coding subspace because BACS maximizes correlation? Here, we first calculate the projection of neural activity onto each of the subspace, and then calculate the correlation between the projections in this example area pair. Here, each row is 1 subspace, and the x-axis is the time. And this is the noise correlation calculated within each child type, and then averaged across child type. We see that although correlation along coding subspace is high, along BACS it is higher. This is not surprising because the definition of BACS is to maximize correlation. But this also means that there is some different axis from the coding subspace that has even higher correlation. And that's why BACS did not pick the CD. This plot shows the first 5 axes found by CCA. We see that the projection correlation along the first axis is much higher than the rest of the axis. And therefore, in the results I'll mention today, we'll focus on the first axis of BACS. As a summary of the first part of the talk, we identified the 3 subspaces, and we found that these subspaces have very different dynamics, different decodability, and different between area correlation. In fact, we also found that they have very different temporal stability, but since time is limited, I'm not going to dive into this analysis. So all these distinct features emphasize the importance of subspace-specific analysis. Now that we have identified and examined the 3 subspaces, we wonder what the relations between the subspaces are and how shall we interpret them. Let's first go back to the definition of the subspaces. As I mentioned before, BACS is defined by canonical correlation analysis, which maximizes correlation. While CD encodes task-related variables and MS encodes movement, it is hard to interpret the meaning of BACS. So before we dive deeper into this question, let me first clarify how we defined the motor subspace so that you'll have a better understanding of what's next. We trained an autoencoder to turn each image frame from the video into an embedding vector. And then we take the embedding vectors and perform canonical correlation analysis between the embedding vector and the neural activity. So the CCA therefore finds subspaces within the neural activity that maximizes the correlation between neural activity and the embedding vector from the videos. Now, getting back to the question of interpreting BACS. A straightforward way to do this is to see how much overlap it has with CD or MS. Here on the X and Y axis, we're showing the dot product between BACS and MS versus the dot product between BACS and CD choice. And each dot is one session. We see that for all the sessions, BACS has higher overlap with MS than with CD. But since BACS maximizes correlation, we also want to see how correlation is affected by coding or motor. So let's take BACS and CD as an example to see how correlation along BACS is affected by coding. We first calculate the CD choice within the neural activity space. And then we remove the neural activity along the CD choice to get the population activity decorrelated to coding. And we calculate BACS again in this decorrelated population activity, and we get BACS decorrelated to coding. Similarly, between BACS and motor subspace, we can also calculate the MS first, and then achieve the population activity decorrelated to motor, and then calculate BACS again, which we call BACS decorrelated to motor. So note that MS can include more than one axis. We actually scanned across the number of MS axes, and the conclusions hold the same. So in this talk, all the motor decorrelation process uses the first 5 axes in the motor subspace. Then we calculate the projection correlation within BACS and all the other subspaces. This is for one example area pair, and we found that while BACS has the highest correlation, after we remove coding, the correlation dropped by a bit, but after we remove motor, or after we remove both motor and coding, the correlation dropped a lot. And this is also true on the single session basis. Here, each dot is one session, and the X and Y axis are the correlation drop in BACS after we remove motor or after we remove coding. So across all the area pairs in this data set, we see the same conclusion. Here, the x-axis is the log 10 of the p-values that compares the correlation drop in after we remove coding versus the correlation drop after we remove motor. And the positive p-values means that removing motor has a higher drop in BACS correlation than removing coding. Therefore, it shows that BACS has more overlap with motor than with coding. But then what about the relation between coding and motor? Because we know that even uninstructed movement of the mouse could reflect the choice of the animal. And therefore, it will be interesting to see whether coding contains internal representation of choice that is unrelated to movement. To investigate the relation between coding and motor, now we use the similar decorrelation method, but between CD and MS. Basically, we calculated the motor subspace, and therefore the population activity decorrelated to motor. And then we calculate CD again to get the coding subspaces decorrelated to motor. Let's again start with the most straightforward dot product. Here, the first row shows the dot product between CD choice and MS, and they are low, indicating that there's a small overlap between the two subspaces. And then the second row shows the dot product between CD choice before and after removing motor. This dot product is high, which indicates that removing motor does not have a big effect on CD choice. This seems to imply that the motor subspace is not very aligned with the coding subspace, as is shown by this illustration. Similarly, CD trial type is not aligned with motor subspace either, as we can see from the dot product values. However, dot product alone cannot answer the question of whether there is some internal representation of choice that is unrelated to movement. And therefore, we decoded choice within the coding subspace at the late delay epoch. Here, each line connecting two dots is one session, so that we can compare the choice decoding AUC along CD choice before and after removing motor in the same session. And after removing motor, CD choice decoding AUC decreased, but a good portion of the decodability still remained. Therefore, removing motor does not abolish coding. If we look at the AUC across time, before the go queue, a fairly good portion of choice decodability remained after removing motor. After the go queue, the animal is making large movements that are correlated to their choice, and therefore there is a large drop in AUC after we remove motor. Across all the brain areas of interest, this conclusion holds true. We found that removing motor leads to a drop in decodability, but it does not abolish coding. To summarize the second part of the talk, we interpreted the relations between subspaces. Specifically, we deepened our understanding about BACS by finding that its correlation is more affected by neuron than by coding. And by examining the relation between coding and motor, we found that removing motor does not abolish coding, especially before the go queue, which indicates an internal representation of choice unrelated to movement. So we just looked into the differences and relations between different subspaces. Since all these subspaces were calculated using the data in control trials, we'd like to utilize the perturbation trials to validate some of the differences between the subspaces. In the third part of the talk, I'd like to discuss the capability in subspaces of predicting perturbation effects. Our dataset contains perturbation trials, where optogenetics inactivation is applied to unilateral ALM at the early or late delay epoch. Simultaneous recordings of subcortical downstream areas allows us to observe the effect of ALM perturbation on the neural activity in these areas. In this example session, ALM is inactivated at the late delay epoch, and we are recording from medulla to see if ALM perturbation has influence on the neural activity of medulla. This plot shows the spike rates in the medulla in control trials and in perturbation trials. We see that when ALM is being perturbed, the spike rate decreased in medulla. And similarly, we can plot the choice selectivity in medulla again in control and perturbation trials. Here, the choice selectivity was calculated as the difference in projections on CD choice between lick right and lick left trials. Now, we'd like to see if these perturbation effects in medulla can be predicted by neural activity along subspaces. In the scatter plot, the x-axis is the correlation along CD choice between ALM and medulla, and they are calculated in control trials. And the y-axis is the change in spike rate in medulla when ALM is being perturbed. This is basically the difference in spike rate between control and perturbation trials. The red dot here is the outlier data point determined by linear regression. We see that the spike rate change during perturbation cannot be predicted by correlation along CD choice because there is no significant correlation in the scatter plot. Now for the x-axis, if we instead use the correlation along BACS and keep the y-axis the same, it still cannot predict the spike rate change in y-axis. Now let's look at the selectivity change in medulla during ALM perturbation. Here again, the x-axis is the correlation between ALM and medulla along CD choice calculated in the control trials. and the y-axis is the selectivity change in medulla. The two p-values are calculated with and without outlier data points, which are not visualized here. And we see that the ALM perturbation effect on selectivity in medulla can be predicted by the correlation between medulla and ALM along CD choice. But if we change the x-axis to correlation along BACS instead, it cannot predict selectivity change in medulla anymore. To summarize, between area correlation along CD choice is able to predict the selectivity change in medulla during ALM perturbation trials. Although this is only one example area pair of ALM and medulla, we actually saw the same conclusion in most of the area pairs of interest. And comparing CD with BACS, although BACS is calculated using CCA, it is not able to predict the perturbation effect in spike rate or selectivity. I want to emphasize again that all the subspaces and the correlations along subspaces were calculated on control trials only, yet perturbation effects can still be predicted. This perturbation analysis shows that even though CD is calculated through maximizing subspace decodability within single areas, and it does not have maximized correlation between areas, the between area correlation along the subspace is still able to predict the selectivity change on one brain area when the other area is being perturbed. Finally, before we conclude, I'd like to quickly show you another analysis on the variability of between area correlation. Let's say for each session, we calculate the between area correlation in subspace 1 and in subspace 2 and compare the variability across sessions. In this illustration, the two between area correlations are correlated, meaning that sessions with higher correlation in subspace 1 also have higher correlation in subspace 2. If we have more than two subspaces, we can summarize the correlation of correlation from the scatter plots for all pairs of subspaces. Note that this illustration is only one example of this correlation of correlation being high. It does not have to be the case in reality. And this is how the matrix looks like on our data set for all the subspaces of interest in one area pair at the sample epoch. While some subspaces are correlated, a lot of them are not. And this matrix also changes across epochs. For example, at the delay epoch, it looks very different. For the sake of time, we're not diving into this analysis, but we found that the strength of between area correlation varies across sessions, and this variation is modulated by specific subspaces, epochs, and area pairs. In conclusion, we first looked into the importance of subspace-specific analysis. We started by defining the three subspaces: the coding subspace, the between area correlation subspace, and the motor subspace. We showed that different subspaces have different dynamics, different decodability, and different between area correlation. They also have different temporal stability, but we skipped today. We then tried to interpret the relations between subspaces. Specifically, we found that BACS has more overlap with motor than with coding. And we also found that removing motor does not abolish coding. Finally, we looked into the capability in subspaces of predicting perturbation effects. We found that CD correlation predicts perturbation effects on task-related activity. Finally, this project is a team collaboration between the Druckmann Lab, the Svoboda Lab, and the Li Lab. I'd like to thank our teammates and funding sources. In addition, I'll graduate this summer, and I'm actively looking for a postdoc position starting this fall or winter. preferably in the San Francisco Bay Area or Boston. So if this draws your interest, please kindly let me know. Thank you for your attention.